Hi, this is Giancarlo Vicente, and this is my third attempt at recording this presentation. This presentation will be about fatty acids in triglycerides, how it proves an effective platform for biological drug delivery. The hypothesis for the experiment was that unhealthy fat could effectively transport oral medication to your body and eliminate the need for some injections or IV treatment. The reason for this experiment was that injections and IV treatments tend to be more expensive than regular oral drugs. Brief explanation of the experiment. The Houston Methodist nanomedicine researchers are studying this new drug delivery system for diabetes drugs that resulted in approximately 25% absorption in mice model, which is considered to be very high for an oral drug. The human body can absorb fatty acids. So we, who were the scientists of the Houston Methodist Nanomedicine Institute, decided to chemically link biological drug molecules to fatty acids to see how well these drugs are absorbed into the gastrointestinal system. The Houston Methodist Nanomedicine researchers were the group that carried out this experiment under the guidance of Dr. Heife Shen, who is a professor of nanomedicine at the Houston Methodist Research Institute. Now, to describe the experiment. After designing a platform where a peptide-based drug used to treat diabetes mellitus was chemically linked to fatty acids, Dr. Shen and the team packaged the resulting combination in a nanoparticle that was resistant to gastric acids in the stomach. Now, once inside the small intestine, the drug molecules were released from the nanoparticle and the mice absorbed 24.8% of the drug dosage. Now, to describe what were the different variables in the experiment. The independent variable was a nanoparticle that was resistant to gastric acids in the stomach. Dependent variable for this experiment was how well the drugs are absorbed into the gastrointestinal system. The control group from the research published was not clear. However, it seems as though it was either the mice that were used or the amount of drug molecules that were released. The result for this experiment is that once inside the intestine, the drug molecules were released from the nanoparticle and the mice absorbed 24.8% of the drug. Now, this is relevant and very important because in the past it's been demonstrated that the absorption of the molecules into the body was not possible given that the drugs used were not able to resist the were not able to resist being decomposed by the gastric acids in gastrointestinal system. The conclusion to this experiment is the following. With this experiment, the nanomedicine researchers have proven the drug platform works in animal models. And the next step is to perform toxicity studies and produce the drugs in a larger quantity and eventually move to clinical trials in patients. The source for this experiment were the researchers' findings, which were published in the Science Advances on April 1st, and the article for this news was published in Medical Express. Thank you, and I hope you enjoyed.